down.
See you. I know she just came up here. But you can tell jokes.
Think loud.
Bellini, where your district is.
Bring the microphone closer to your face if you think you need it.
I'd like you to comment. Hey, thanks for boosting the facilities.
So which one did you help elect?
Yeah, well, this is Melissa will let you today.
Good morning. The House Energy Policy Committee will come to order. Clerk, please call the roll.
Chairman Glenn?
Here.
Representatives Hout? Barrett?
Here.
Cole? Tedder? Molino?
Here.
Farrington?
Here.
Griffin?
Here.
Johnson?
Here.
LaFave?
Here.
Lauer?
Here.
Riley?
Here.
Lozinski?
Here.
Zianda? Garrett? Camilleri? Elder?
Here.
Green?
Here.
Sneller?
Here.
Mr. Chair, you have a quorum?
Thank you, Mr. Clerk. Representatives Cole and Tedder have joined us since you called their names. I need a motion to approve the minutes for last meeting. Representative Tedder so moves. Without objection, so ordered. Today, we are going to address nuclear power and the effects of it. And happy to have with us today for the next 90 minutes Dr. Eugene Moore. I've had the privilege of visiting Dr. Moore's home in Midland over 40 years in the chemical industry, and his home is decorated with trophy after trophy, patent after patent. Can't keep up with all the awards that he has won in terms of chemical engineering, primarily at Dow Chemical. And also a compatriot of his, Dr. Jerry Cutler, who is a nuclear scientist and expert on the beneficial effects of low-level radiation. Now, maybe that gets your attention right there. But I think one of the things we're going to be told today, yes, right, as Representative Barrett notes, these are smart guys. They know more about this than we do. They're smart meter, that's right. And I think they agree with me on smart meters too. But anyway, that's a different issue. But Gene, are you going to start off?
Yes.
Okay, you need to push the red button at the base of that microphone. And we will start with Dr. Eugene Moore, formerly of Dow Chemical. And then Mr. Cutler has a presentation, some slides. You have a copy of it at your desks, and then we'll open it for questions. So, Dr. Moore, if you would proceed, please. And welcome.
Thank you. Thank you. It's nice to be here. I'm concerned that, or Jerry Cutler's concerned that my introduction might be so long that he won't get a chance to give his presentation. But I'm trying to make the point in my introduction that nuclear power could be tremendously less expensive if we realize that low levels of radiation are actually beneficial. And Jerry will make that point during his talk. And I'm going to try to go fairly quickly through the set of introductory slides here. Going back to 1945, there was a series of articles, double-page big articles that talked about the broken atom in our destiny. And the word back then in 1945 was long sought and incredibly vast new source of energy, now released, promises permanent peace and infinitely better life for everybody. And just a couple quick flicks of that. And the question is, what happened? What happened? Why didn't those dreams come true? And why is nuclear electric power too cheap to meter? Combination of vastly overregulation, greed, and false cancer fears. And why shouldn't low-level radiation again be used in medicine? It was used widely from about 1902 to 1952 when antibiotics were introduced, and there are thousands and thousands of references. Gas gangrene was perhaps the first one where it would progress at about an inch an hour, and the only cure for it that was known at the time was amputation quickly. And I want to make a quick point on power cost driving industry. A few years ago when I was at SRI, we were doing industrial analysis, and the Gulf Coast electricity was 3.5 cents per kilowatt hour. And I recently checked on Midland electricity on my own bill, 13.9 to 16.3, and you have to ask what industries would move to lower cost electricity, and if we could make nuclear power available at low cost in the U.S., in Michigan in particular. And just a quick word on complexity of the simple nuclear plant. I was involved in a chemical process to make tertiary butyrylstyrene. We had a very complex reaction, and we operated with 15 total people in the plant. And recent publications on Palisades says they need 500 to 600 employees to operate the plant. And I think a lot of that is due to overregulation. And I'm not going to go into the details of this, except I was pretty close to the Midland proposed nuclear power plant back in 1960s. That was originally forecast to cost $272 million, and it ended up never being completed and costing $4.5 million due to the regulations that simply invited greed. And a very interesting book that Jerry has brought to my attention is Underexposed and What If Radiation is Actually Good for You. And quick overview on Dr. Cutler, doctor's degree in nuclear sciences, active since 1971, past president of Canadian Nuclear Society, and very much published on health benefits of low radiation, and he's lectured very widely on these benefits. And the final question is, what if Michigan could realize the inexpensive nuclear power and perhaps have cheaper power than the Gulf Coast, and Michigan would be a leader in, what if Michigan could be a leader in the application of low-level radiation? And I'm not going to say much more. I'm going to turn the floor over to Jerry Cutler.
Thank you.
Thank you, Dr. Moore.
You can see my background was mainly in nuclear energy. I worked on Kennedy 6 reactor at HP. I'm an engineering manager as well. And
Mr. Cutler, could you move?
Health effects of radiation.
Mr. Cutler, could you move over toward the mic? Move your chair over toward the mic there, sir, please.
Sorry.
Thank you, sir.
Try to switch this off and put on my slides. Sorry, I'm not... Okay, this is my presentation. Go back.
Dr. Cutler, do you want us to ask questions as you go along, or do you want us to hold the questions until you're finished?
Well, I've got 82 slides, so if you want to ask me a question, it'll take some time to answer, but unless the question is really, well, feel free. I don't want to stop you from asking questions. Just take into account that if you want me to finish in 90 minutes, we have to keep the discussion short.
Yeah, and that means your slides will be less than a minute each, so we do
I'll
have
try.
time.
Some will be less and some will be more,
Okay.
yes.
Thank you, sir.
Okay, Madame Curie, you've known her, she's very famous. She invented the word radioactivity, and she discovered radium and polonium, and won 2 Nobel Prizes. And her point is nothing to be feared, just try and understand what's happening. You all know about Fukushima. This is the accident. The red part shows the higher radioactive contamination that was released. I did some arithmetic. See if I can get this to work. This is the hot area. This is the highest radiation in that area, 91. If you take that, multiply by the number of hours in the year, you get a total dose of about 800 millisieverts per year. This is natural high background radiation in some areas. So even the highest area and the highest point, assuming it was the same over the whole year, didn't decay, and again, it's the highest point, most of it was less, there really wasn't much more radiation than what occurs naturally in the world. Now, if we want to get more specific, this was an analysis done by the Japanese energy. This is the high area is point A, and I can get this pointer to work.
Dr. Cutler, we need you to speak into the mic if you don't mind, please.
Okay. Point A is the highest area there, and if you look at the integrated dose over a year, it comes out to, in the table there, 235 or something, or 200, and the highest level in the city of Ramsar, 35,000 people, is 260. It's even higher than the highest point. Bottom line is they evacuate a lot of people. So what happened in Japan in 2011? 20,000 were killed or missing due to the earthquake and the tsunami. 300,000 people were evacuated because of the fear of the radioactive material that came out of the damaged plant. 1,600 old people died prematurely because they were pulled out of homes and ICU and were put in auditoriums, evacuation centers, sat on a floor in a cubicle for 5 years. It's incredible. And the interesting thing is no one was harmed by radiation, not even the workers in the plant. And so this fear of radiation, well, it stopped the Japanese reactors, 50 of them. They started importing oil and coal, and well, it was a huge economic disaster, it was a social disaster, and the fear spread all over the world, and people in California were terrified of the fish and the radioactivity that was coming on the other side of the ocean. So it's unbelievable how much fear is generated by radiation. So, is it safe to be exposed to radiation?
Well, Dr. Cutler,
Yes?
Representative Barrett would like to ask you a question, sir.
Please.
Sorry, I apologize for interrupting your presentation, but I was curious, is there a way to know at the moment that this exposure is taking place if it's a harmful level or not? Was there an overreaction, or was there just an unknown and erring on the side of caution
We've
reaction?
known what happened in Chernobyl, and that was a big
Right.
accident. That's the reactor blew apart, the top was open, it was spread all over Europe. I mean, that was big accident. Nobody really got overexposed, not even the plant workers. The only people that got harmed were the firefighters who
In
were
Chernobyl?
standing around the opening trying to put out a nuclear fire.
Sure.
You can't put out radioactivity with water. So the graphite was hot down there, and the radioactivity was producing the heat, and they were pouring water on it, and those firefighters got overdosed. There's 134 were hospitalized, 28 died within weeks, and 106 recovered. The cancer incidence of those people was no greater than the cancer incidence of normal Russians, and the mortality also comparable. So although there was enormous fear, there was fear of thyroid cancer in children's. They did all this screening, they did surgery on the poor children, took out their thyroid glands. It was unnecessary.
Okay.
So
Thank
that's a
you.
long answer.
No, I appreciate that. Thank you.
So what happened here was pure fear. That's all because of regulation, and well, I'm going to get to the problem of the fear. So the basic problem of nuclear energy is that people are afraid of nuclear plants because we tell them that any radiation exposure will increase the risk of fatal cancer. This is a 1956 anti-nuclear health scare, and it's false. The low radiation, or low radiation dose rate stimulates adaptive protection system. It doesn't cause cancer. There's 150 genes in humans that that operate your adaptive protection system. So the effect of radiation on health is high radiation exposure inhibits or damages these systems, and low doses stimulate them. And for every type of exposure, there's a threshold. A dose below is beneficial, a dose above the threshold is harmful. And really, instead of talking about cancer, we should be talking about longevity. This is the best measure of the health effect, not cancer. Cancer was created to make a scare, to stop bomb testing. Let's look at cancer, the death rate. You'll see that it rises exponentially with age. At about age 40, it starts really going up. The top graph is a linear scale, and the bottom graph is a log scale. And you can see it's a straight line, it's exponential. So where are these cancer cells coming from? They occur spontaneously. This is damage in DNA from free radicals, reactive oxygen species, thermal effects. Where does that reactive oxygen species come from? It's because we breathe oxygen. The oxygen we use to breathe them to consume glucose that's transported in our blood. Our bodies eat, turn it into glucose. The glucose goes around to our cells, the brain cells everywhere, and when our blood also carries oxygen. The oxygen reacts, gives us energy, we can operate. But the same oxygen damages the DNA, it damages all the cells in our body. So why do we live? Why do we live 70 years, more, 85? It's because we have protection systems, and they keep us alive from birth until the end. The problem is, this increase, it's our protection system age. They get weaker, immune system gets weaker. And if you want to do anything about it, low-dose radiation and other things stimulate immunity, stimulate our protection systems, and that will keep us healthier, and it will stop or delay the cancer incidence.
Dr.
Any
Cutler, Representative Lefebvre has a question.
Yes, please.
Thank you, Mr. Chairman. My question is, in the 1970s, we used and prior to that, we did a lot of nuclear testing
Yes.
with ballistic missiles, and during the Cold War. Has that, in your opinion, does that have any effect on the cancer rate generally because we blasted off a lot of these bombs?
No, as I mentioned, those released low-dose radiation to the surrounding population. Low doses stimulate protection systems, so there was no harm. If anything, it was benefit. Maybe not enough benefit that we should get, but certainly it was not harmful.
And
So any reason for compensation for cancer that may have occurred later was inappropriate.
and that background radiation, is that the type of radiation that we see when we try to carbon date things after 1970?
Yeah, that would be carbon-14 dating. Yes, that happens naturally. A lot of it happens because of cosmic radiation reacting with nitrogen and turning it into carbon-14.
Thank you.
Okay, so we talked about cancer a bit. As I mentioned, cancer immunology, low-level ionizing radiation can play a key role in stimulating immunology. And in that article was published this year, 222 references. So there's a lot of information out there on low-dose radiation and how it stimulates our protection systems. This is what a radiation, and also for most the chemicals as well, a dose-response model. You see on the abscissa at the bottom, the dose or the dose rate is increasing to the right, and the health effects going upwards on the ordinate. And you'll see where the control group gives a normal incidence of any illness, including cancer, and at some threshold, the exposure stimulates beneficial effects, and then there's an optimum level, and then as you keep increasing the dose or the dose rate, you pass a threshold. NOAEL is called no observed adverse effects level. So it's the same level as the control group who are non-exposed, and when you go beyond it, that's where it becomes harmful. So anything in the region below the threshold is not harmful and beneficial. So we used radiation in the old days. Remember, this was discovered in 1895 by Röntgen. So from early 1900s until 1960s, we were using radiation to eliminate cancer metastases and slow cancer growth. We used it to accelerate wounds healing of wounds by stimulating your protection systems. We stopped infections, including gas gangrene, carbuncles, boils, sinuses, inner ear, pneumonia. You just name a lot of infections, and if you stimulate your protection systems, they will get cured faster. A lot of these didn't have cures in the early days. There were no antibiotics, and they were using low-dose radiation, and used it to treat arthritis, to relieve the pain, reduce inflammation, treat swollen lymph glands, and cure asthma. Actually cure it. People with asthma would come to a doctor. The doctor would take an x-ray to see how much congestion in the chest, and the next day the patient came back and says, "I'm fine, I don't have asthma anymore." So it happens within hours, days. Now the new discovery that we'll talk about is you can actually reverse Alzheimer's and Parkinson's disease. And Eugene's wife has just been treated with low-dose radiation. She's made a recovery of partial of Alzheimer's. She eats and she's responsive. And Eugene has Parkinson's, as you may have noticed, and he's had the same treatment, and the tremors during his sleep have disappeared, and his dose that he takes of chemical pills to relieve the symptoms, he's taking fewer pills. So we have people in Michigan who are already using modern. So this is a new discovery that I'm doing studies in Toronto with, because I can't interest anyone to do that here because doctors don't believe that radiation and low doses are beneficial. So how did the linear no threshold model happen? Early geneticists, Hermann Muller, discovered he zapped fruit flies with huge amounts of radiation, and he discovered that germ cells are mutated, and at very high dose and at high radiation intensity, the number of mutations was proportional to the dose. Now, another scientist, Ernest Caspary, used low-dose radiation and lower dose rate, and he found a threshold. He saw that the mutation rate in the exposed flies was the same as the unexposed flies, the controls. And so he noticed already that there was a threshold at a lower dose. Muller ignored the Caspary evidence, and he proclaimed, he got a Nobel Prize for discovering that X-rays cause mutations. And he said there is no escape from the conclusion that there's no threshold. So he actually lied in a speech at the Nobel Prize lecture, and he was trying to create a scare of radiation. Part of this was due to the atomic bomb explosions in Japan, and they trying to discourage both Russia and U.S. from developing more weapons and the arms race. So he was trying to accomplish a political goal by misusing science. The genetics panel of the National Academy of Science, the U.S. National Academy of Science, the Beer Committee was biological effects of atomic radiation. They recommended in 1956, as a result of Muller's work, that should use a linear no-threshold model to assess genetic mutation risk. Now, when you talk about genetic mutation, that leads you to the idea of cancer. And the other problem is they were using, even though he was talking about radiation, they applied this also to chemicals. So any amount of a harmful chemical is toxic, and you can project the risk all the way down to zero using a linear no-threshold model. And the whole issue is, do you get more benefit from the chemical versus the risk? And they always keep talking about risk. But if you go back to the model I showed you earlier, there's a low dose range where it's beneficial, every chemical, and above the threshold it's harmful. So this is the proper biological interpretation of exposures, and this was all perverted by this recommendation of the National Academy of Sciences. And so regulators all around the world, everyone, adopted the linear no-threshold model to assess cancer risk for normal cells, and there is no evidence to support that. So it's all being done on the theory of the model that everyone accepts, and all they're trying to do is be cautious and conservative. This is a renowned professor, Ed Calabrese. He did a study for more than, well, almost 20 years on what happened, how this linear no-threshold model came about, and his paper last year is called LNT Gate, reference to Watergate. The scientific misconduct by National Academy of Sciences led to governments adopting the LNT model for cancer risk assessment. So this was last year's paper. This year, he's got another one, the ideological history of cancer risk assessment. And if you can read the fine print, which I'm having trouble reading myself, it's basically talking about an ideological driven deliberate falsification, fabrication of the research, and the recommendation to change from using a threshold to a linear no threshold model, and misleading the entire world. And so he's trying to expose this. The problem is people are not paying attention to this because they're accustomed to what was being done all these years. Looking at what Herman Muller did, Japanese repeated the study in 2009, and they used cobalt-60, same kind of fruit flies, and the results they got show that at high dose, the original data are the black dots. The way over to the left is, you see the dot at 0, that is the natural occurrence, the spontaneous mutation rate. You see it's 0.032, and if you look at low dose, it's actually 3 times lower, the mutation rate of those fruit flies. And so the original data is in black. I produced the blue dots by pooling the data on the 2 neighboring points and getting a more accurate, less variable. And what you see here is there's a threshold at 1 gray where the effect of radiation becomes harmful. So you see above that dotted line there's risk, and below it is benefit. And the linear model is shown as a green line, always showing harmful effects, whereas the blue line shows significant benefit in the low dose range. So this was repeating the study that was done by Muller, but Muller used 1,000 times higher radiation level, and this is in the low dose range where we actually get exposures. And here you can see that the low doses that we would be exposed to, even in an accident, are beneficial even for fruit flies. Now, we shouldn't be talking really about cancer. We should be talking about longevity. It's the best measure of health effects. The radiation scare, any dose increase causes cancer risk. Cancer is an ideal anti-nuclear scare. Cancer is very complex, has many causes, many confounding factors, not well understood. It's difficult to predict, and everybody dreads it. So it's an ideal scare if you want to produce a radiation scare, is to say it's causing cancer. But if you really look for the best measure of the health effect of radiation, it's longevity. There was a renowned scientist in Wisconsin, John Cameron. He did a study where he looked at the data from the early radiologist, and for the nuclear shipyard workers, there's another study. There were many studies, but in those two in particular, he showed that low doses of radiation actually produced a benefit. This is human data, and this has all been ignored. The nuclear shipyard workers was paid by the Department of Energy. $10 million was a lot of money back in the time when they did this study. And then they never published the data because they got the wrong answer. They were looking to see more risk, and they found the opposite. Calabrese and Baldwin looked at the evidence of a lot of studies. Remember, we've got 120 years of data, studies being done by scientists. They saw increased lifespan with low doses in mammals, increases of 10 to 30% versus the unexposed ones. Radiation, again, stimulates adaptive protection systems. At low dose and at high dose, it inhibits it. And the spontaneous, it's acting against spontaneous cell damage. And as I mentioned, it's caused by breathing oxygen. And so it acts against that and any damage that's caused by other causes in addition to radiation. So the main effect of low doses is reducing the damage. It's oxidative damage. This is the mortality of the British radiologist. You'll see before 1921, this is before they had any protection at all. If you look at all neoplasm, which is cancer, the observed, the O, is higher than the expected, which is 43. And you see observed divided by expected, you'll see is 1.44. There's an increase in cancer incidence. But if you look after 1920, this is when they had protection standards in. The observed was 72, the expected was 91, and it was actually a reduction in cancer. And if you look for mortality of all causes, including cancer, you'll see that after they introduced protection levels, the radiologists who got low exposures had a very low observed versus expected mortality. So this is an example of a British radiologist in a study. It was published in 1981 by Smith and Dole. The nuclear shipyard workers, you can see there were 0 dose. These were the controls. These were the non-nuclear workers in the naval program. And then you can see two levels of exposure. And if you look at the above half a rem, you'll see that the mortality is 76%. That's 6 standard deviations accuracy, because we're looking at 30,000 workers versus the controls, which is another 32,000. And you can see there's good statistics there, and it shows in human data that there's a lower mortality, and the study was not published. We have a report from Los Alamos, 1995. He's a renowned scientist there, Inkret et al. And they talked about the history of radiation protection, and the tolerance dose rate. This is the exposure levels that was permitted. And you can see in the 1920s, they had a level of 70 rem per year, which was acceptable dose rate that was allowed for radiologists. And then a few years later, they dropped it down to 50. You'll see that on the left side, it's exponential. It's a log scale versus year on the bottom. And you'll see how the radiation protection kept being ratcheted down really dramatically. And finally, in modern times, it's several hundred times lower than what was acceptable in the time that we had 30 years' experience with radiologists. And then the 1950s, when they brought in the radiation scare, you'll see there's a new red line there for the public. And apparently, it's acceptable to zap the radiologists, but not the public. Well, gee, they're all people, and you got to protect everybody. So they had two standards, and even today, what's allowed to the public is a factor of 10 lower than what's allowed to workers. And really, there's no justification. Everybody should be able to get the original exposure of 70 rem a year, because that was established way back as a safe level. This is a paper that I published earlier this year. I've done analysis on dog studies, and this is a study that was done on various mammals. It was published in 2012. The blood system is the most sensitive system, the most sensitive cells. The bone marrow are most sensitive, and the first cells that react to radiation are well, the first cancer issue has to do with leukemia. So the blood system responds to chronic radiation, and in this study they looked at 10 accidents for humans. They looked at studies on rats, on dogs, and what they found was radiation effects of function of dose rate and total dose. Stem cells are very radiosensitive, the blood ones, but they tolerate and adapt to radiation. And they adapt better if the dose rate is low. And so what happens when dogs are exposed, their cells mutate, but they still maintain a service of blood function. Beagle dogs, the lowest that they had, there were 10 groups that were studied. The lowest ones had the same cancer rate as the controls, and that low level was 250 above background. So what they found in this study, in this paper, was that the original levels that they had in the 1920s, 30s of 70 rad per year is still okay, and yet the present regulations are not justified. So this is a study that was done on 10 different groups of dogs. And the first group, the very high exposure, you can see the dogs lived only about 25 days, but then as different groups of dogs, there's 20 dogs in each group. These are mortality curves, and you can see I drew a line across at 50% mortality, it's sort of the median, and then I drew a line across at 10% and at 5%, and I looked at the intersections of those lines against the mortality curves, and I took off data, I tabulated them, and I analyzed them, and I plotted a curve. This is analysis that I did in my paper of data on dogs that was produced by the Department of Energy. And what you can see is there's a range from the controls with normal background, and the first data point is way above, well, it's 200 times higher, more. And that's when the lifespan begins to decrease. And the black dots are the median. The 5% mortality, this is the tail end of a delta function, a bell curve. And what you see, and what I see in this data, is that the blue dots are short-lived animals, and they get the biggest benefit of low-dose radiation. They seem to live about 30% longer than the controls. And even the medians see a benefit of about 15%. And look how quickly the lifespan decreases when you get to very, very high dose rates. That's the log scale on the bottom there. But this whole range from the controls to the first data point is not harmful, and yet everybody's predicting cancer risk or lifespan effect, health effect, negative effect. Now, I did the same review on dogs that inhale plutonium. Plutonium, everyone recognizes, a terrible substance, the most toxic, yada, yada, yada. All these anti-nukes are telling you that. Well, what they did was they took plutonium dioxide, they ground it into an aerosol, and they got the dogs to inhale it, and this plutonium stays in the dogs for their entire lifespan. It doesn't breathe out. You don't exhale it. It sticks, and it irradiates the nearby cells. And so, again, we have, these are longevity curves, survival curves, but again, I did the same kind of analysis. I looked at, you can see the red circles. You can see the dogs in the first group that got a little bit of plutonium saw a significant increase in their lifespan from the controls, which are the 0 line, and 1 is the first group. And the same thing happens at 10%. And then I've got another line across where I took the intersections off and plotted the same kind of curve. And those are data points in the actual low dose range, and you can see a significant increase, like 50% increase in the lifespan of short-lived dogs. And the red are the 10% part of the bell curve, and the black are the median. There you don't see much of an improvement due to plutonium, but it's remarkable there's an increase. But then, of course, when you hit the high dose, as I always said, high doses are harmful, they cause damage. The interesting thing is these plutonium particulates are in the lungs, and they only irradiate the nearby cells. They only have a range of 1 or 2 cell lengths. And yet this is affecting the whole lifespan of the dog. So it's not causing little tumors where the plutonium particulates are. It's sending signals. It's signaling protection systems that's increasing the entire lifespan of the dog. So the whole model that they had of radiation hitting cells, causing mutations, causing cancer, it's all wrong. The cancer is being caused by the oxygen we breathe. The radiation is affecting our protection systems. Low doses stimulate them, high doses inhibit them, and signaling is a big factor. You can have radiation in one area and it'll send signals to other areas. This is another study of inhaled plutonium.
Dr. Cutler, let me interrupt you just a second. Representative Griffin has a question.
Please.
Thank you, Chair. Actually, I have 3, but they're short, and it'll help us understand the significance of the study. First, what was your sample size for the radiotoxicity study?
The dogs?
Yeah.
Well, there are 20 dogs in each group.
And how many groups?
Well, one of them had 10 groups. The cobalt-60, the gamma rays had 10 groups, and the plutonium had 7 groups,
Okay. And then...
including the controls.
Perfect. Okay. So then, I'm sorry, the font is too small on this, and I can't see it up there.
You have a handout,
What was the...
but-
Yeah, I know. I just
Okay, sorry.
forgot my glasses, so I can't read it.
That's the same problem.
Sorry, but what was the item? Oh, look at that. What was the date of the published study, the radiotoxicity one?
The cobalt-60 was 2012. It's a 20-year study, and the paper I got was 2012. I can give you references. I can send you actually PDFs of the papers. And the plutonium study that I used there was 2008.
Okay,
They
and
were
then-
done out west at Hanford, and there's another, in Albuquerque, there's another place with studies inhaled particulates, radioactive. So these were DOE studies. The reason they use dogs is they're a very good simulation of humans, much better than mice or fruit flies. And their lifespan of 15 years allows you to do a study in a 20-year period, whereas if you can't study humans, but you can study dogs because you can control them very well. You know, there's always, when you talk about cancer, there's hundreds of confounding factors. Well, maybe less, but a lot of confounding factors. But when you work with dogs, you can control their exposure, you can control the temperature, you can control the food they eat, you control the genetics. You breed the dogs, and they're all the same. So the only thing you're looking at is the effect of the radiation. So dogs are very good studies, and they're done very well by the US DOE scientists.
Thank you. Thank you very much for that. And then one final follow-up for you, if you don't mind. Have you done a meta-study of other studies to compile that simulate this? Is there anyone else that's replicated this in the same
Yes.
time frame that you have? Maybe just a quick comment on that would help us put this into perspective, because, I mean, are you the only researcher that is compiling this, or just give us an idea of the scope?
Well, you have hundreds of scientists studying dogs. There's, what's his name? I have several friends in the Department of Energy who've been doing studies on dogs. They've been publishing in the Health Physics Journal, and I've been looking at all their studies, and the studies show the same thing. And they've been publishing, and some of these, they're not very popular, these scientists. Otto Rabi, you want to write that down? O-T-T-O, Rabi, R-A-A-B-E. Very renowned scientist, did a lot of studies on dogs, on alpha radiation, and he's got similar results. The reason I picked these particular ones is they had a nice set of curves of mortality or longevity that I was able to take and analyze. Because the work I did was to analyze studies that were done by scientists. I don't have dogs. I didn't do the study. So these are very expensive studies. And there are a lot of them. The U.S., we had a lot of workers with plutonium, as you can imagine, in Los Alamos and other places. And they need to know what the effects of plutonium are on humans. So they did a lot of studies starting in the 1960s that went on to the 1980s, and then they analyzed the data, and then they published them, and different people looked at the data again and published it different ways. This is another study. This is inhaled plutonium. You can see the controls. They have a lung cancer incidence of 18%, and the red line shows the other groups of dogs, and you can see the first group there is showing a threshold. Well, it's actually 0 cancers at a very low dose, and I was able from this red line to pull off a threshold at which the lung cancer incidence is the same as the controls. And then I tried to do something clever. I tried to work out how much radon would be equivalent to the plutonium damage, trying to use information from the ICRP, trying to equate alpha exposure from plutonium and radon to sieverts, and trying to work out radon concentration in the homes. This is the data on this graph. Bernard Cohen, who studied 1,661 counties in the U.S., and you can see that as the radon increases along the bottom, picocuries per liter, the cancer incidence goes down, and of course we didn't know where the bottom was, so I was trying to develop a point threshold where lung cancer goes up due to radon, and I got a point way out there based on plutonium data that I was able to pull off. But there are places in the U.S. where the concentration of radon in homes is 10 times higher than that NOAA. So, and people are not dying from cancer there. So I think this point is not very accurate, but that's what I was able to draw based on analysis using ICRP, International Committee on Radiological Protection methodology. Now this is another study. This is instead of plutonium, they're looking at beta gamma emitting radioisotopes. This is strontium-90, cerium, yttrium, 91, 90. These are all beta and gamma emitters. So they mix this into a clay, they grind the clay up into a powder. Again, the dogs inhale it, and you can see the controls. The lung cancer was 15%, and the total body, all cancers in the body, is 50%. And the interesting thing, the first point shows a reduction in the cancer frequency, and even you go up to very high doses of lungs. Here's going up to 25 gray, which is humongous. In the lung cancer, it doesn't even exceed the controls. And on the total number of all cancers, it bounces around a bit, but it's surprising that the linear model that everyone's worried about, it's not shown here in the data on these dogs. Can I jump to humans now? Let's look at Hiroshima, leukemia. This is ground zero, and if you start moving away from ground zero, zone A, B, C, D, E, this is different distances away. There's 3-kilometer line is between D and E. The U.S. Scientists are able to calculate what is the dose at different distances away, and the UN Scientific Committee on Effects of Atomic Radiation. Their first report came out in 1958. There's table number 7 showing leukemia incidence after Hiroshima. Now, what they did was they waited 5 years to let the latency of leukemia build up, because cancer doesn't happen immediately. There's a latency. And they looked at 8 years to get sort of a peak of leukemia versus naturally occurring. And you can see the number of the distance, the dose in those different zones, the number of people in the zone A and zone B. A lot of them were killed by the blast and by the heat of the nuclear weapon. But there's 20,000 people in zone C, 30,000 D, and the E they use as the controls. That's 3 kilometers and farther. And you can see the number of leukemia cases, and you can see 9 is quite remarkable, and that's the natural incidence. But the interesting thing is zone D is actual reduction in the cases of leukemia. And then there's the standard deviation, and then they worked out, this is not my results, it's a photocopy of the UNSCEAR report. And there's a note on zone C, 50 rem. You see there's a superscript there. They've noticed that the people there got very high, severe pain from the radiation, and so they believe that the radiation level they got was higher than 50 rem. So the interesting thing is these people plotted this data, they tabulated this data, but nobody plotted it. So I did. In 2015, I decided it was time to draw a graph. And when you do a picture, it shows something quite remarkable. On the bottom is dose, upwards is the leukemia incidence, and at the lowest dose, the controls, you can see that's the normal incidence of leukemia in Japan. And as you go up in radiation, you don't get a straight line showing linear model showing increase in leukemia with radiation. You actually get a dip. People will argue about statistics of that data, but what is clear is there's a threshold at about 50 rem, or 500 millisieverts, before you see an increase in leukemia. So I published this paper with James Welsh. He's a radiation oncologist, president of the American Association of Radiation Oncology. And I can get you this paper, and basically I'm showing people that, hey guys, the most sensitive cells, your bone marrow, leukemia, the first cancer that happens, you already have a threshold of 50 rem. So why are you scaring people with 1 millirem? Or 10 or 100 millirem, or normal background? There's a huge, and this data was there, it's been there, nobody's looking at it. And when I published this, I got 8,000 hits in the first 2 months. The paper was published. So people are talking about it, but you don't see anything in the news yet. And I looked back at a study that was done in 1957 on the same kind of data, and what I found here was the scientist Ed Lewis. He combined the data in the low dose range, and when you combine the data, you hide the beneficial effect. Do you understand? If you average the data in the low dose ranges, you get 1 point and you don't see the dip, the J part. So that was the sort of thing that was done, the tricks that epidemiologists did in order to hide beneficial and show that this stuff is dangerous at any level. I'm going to, this is, I quoted from his paper where he actually admits that he averaged data. The next slide, I'm going to jump to cancer. And this is a renowned friend of mine, Dr. Sakamoto in Japan. He actually lives near Fukushima in Sendai. And he did studies way back in 1975 showing the effect of radiation on the immune system. He injected cancer cells into the tail vein of mice, and he gave them different exposures afterwards. And what he found that in the low dose range, from 0 to a half of gray, 500 milligray, and that you actually get a reduction in the number of lung colonies of cancer. This is showing that the immune system is stimulated in a low dose range, and above 50 or 0.5 gray, immune system is impaired, is harmed. So there you can see a threshold, and you can see an optimum there.
Dr. Keller, can I ask you?
Yes.
Just summarize a point. All these low dose ranges, are those the ranges of radiation that result from nuclear plants? I mean, the nuclear plants
Are way, way down. They're almost zero. They're almost background. Nothing's coming out of nuclear. We have such great regulation, over-regulation, protection. It's actually safer to stand beside a nuclear plant than it is in other areas where there's natural radioactivity.
Well, I know when we toured the plant down at Fermi, I believe it was one in Monroe, they said that if we jumped in the pool with the radioactive waste rods, that we would be less exposed to radiation than we were standing up in the air.
Yes, that's true.
Now, they said we
The
wouldn't want
shielding
to,
of the water.
we would not want to dive down and touch the rods, they said.
Yeah, but
But at the top of the pool, they said you'd be getting less radiation
30 feet deep,
in the waste pool than in the normal air.
yes. We do over shielding. In the U.S. Navy, the submarines, they've got excellent shielding. The sailors, the submariners get less radiation when they're in the ocean than they do on the surface. And our nuclear plants, the parking lots, everything's paved, so the radioactivity that's coming out of the ground is shielded, so you actually get less radiation around the plant than you do when you get over to this areas where there's topsoil. So this all being measured by airplanes. We don't have a risk, but everybody keeps talking about risks of nuclear energy. Well, I'm just trying to give you a flavor. And these radiation doses are much, much higher than what you'll see in the environment. So, and that's optimum beneficial. So my friend Dr. Sakamoto decided to treat patients with low-dose radiation. This is a source patient scheme where he does half-body low-dose radiation. And he used this treatment to treat a woman who had advanced ovarian cancer, metastases everywhere. And by applying 15 centigrade, which is the optimum he found from the mouse study, twice a week for 5 weeks, which is quite a lot of radiation, 150 rad, all the cancer metastases disappeared in this woman. So he was able to treat the woman with advanced ovarian cancer, remove all the cancer. This is a study that he did on non-Hodgkin's lymphoma. You can see that the conventional treatment with chemotherapy gives you 50% cure rate, but if you lose low-dose radiation, according to that scheme, you get a cure rate of 84%. 15% do not benefit from that. This is a friend of mine who has Hürthle cell cancer. This is a thyroid cancer that was removed surgically, but its Hürthle cells do not absorb iodine, radioiodine, so it's incurable. So he had metastases, and 15 years later, the tumors were growing in his lungs. He was getting the low-dose radiation that Sakamoto used, and he was able to shrink the tumors and control it. So he died of a heart problem, but he didn't have cancer. Now, here's a treatment that my wife got. She had uterine cancer. It was discovered in a pap test during an annual medical. That's Dr. Sakamoto sitting with her. He prescribed the treatment. It was given in Toronto, 150 milligrade twice a week, 5 weeks. And that was done in 2011. We're now in 2017, 6 years later. She's still a happy woman. And when she came out of each treatment with this low-dose radiation, I said, well, how does it feel? And she said, I didn't feel a thing. It's nothing. And so she got this 15 times. Twice a week for 5 weeks. She got it 10 times. And we're very confident the cancer will not reoccur. Now, this treatment is not to treat cancer. The cancer was removed in the surgery. This treatment was to prevent recurrence due to metastasizing that we thought might occur because the tumor was bleeding.
Dr. Representative Barrett has a question.
Yes.
Thank you, Doctor. Over here. Sorry.
Yes.
How does that differ from the conventional radiation treatment for cancer presently? I think that's a more targeted and
Yes,
focused.
exactly. What they do is a high dose, they burn the tumor.
Okay.
And they rotate the machine around so the healthy tissue around it gets less radiation, and the focus gets the intense radiation. So what happens is the tumor dies, the healthy cells recover. The healthy cells have good repair systems. They recover after some inflammation. And then the issue is, have I got all the cancer cells? There may have been cells that escaped from the primary tumor in different parts of the body, and that's the ongoing concern. So many people get treated, and years later, it comes back.
Okay, so then, I guess, by the method you're describing,
It's stimulating
it would be
protection.
more broad or what have you?
Yeah, it's distributed, half a body,
Right.
or whole body.
Sure.
So, and it's low dose, like it's 20 times less
Kinda like a,
intensity than what's given to the tumor.
Kind of like a lamp versus a laser, something like that?
Yes, exactly.
Okay.
And the laser's
I'm a
rotating.
political scientist, so I can relate. We're both scientists here, so.
Well, we use lasers to burn steel. We can actually, those pretty high-intensity beams.
Sure.
Well, they optimize the beam to destroy the tumor without damaging healthy tissue. Now, I want to
Thank you,
show you
Doctor.
some data that this was published earlier this year in February by my colleague Suji Kojima. He did a study on recurrent prostate cancer. So after the surgery, you see there's a line there showing operation, timeline, and then the PSA goes down, and then it comes back because it's metastasized. So the PSA goes up again, and now he's giving low-dose half-body radiation. And look at the PSA go down. It goes down very sharply to close to zero. And then if you look at the metastases, there's arrows there showing tumors all over in the bone due to the prostate cancer that spread. This is advanced prostate cancer. And you can see months later that all those tumors disappeared from the metastases, the bone tumors. So
Doc,
this
you
is
and I,
powerful treatment.
you and I are going to have a talk when we're done here.
I'm here. This is Su Shen Lu, famous Chinese scientist. He came to lecture. He has a son that lives near my home, and he asked me to invite him to give lectures so he could visit his son and grandchildren. So I took him around to various places, including my local hospital, and he gave incredible lectures there, and that was the basis for getting my wife treated. And my other colleague treated with low-dose radiation was they heard these lectures and were surprised. We talked about your protection systems. This is your immune system, and look how it's killing a cancer cell. It grabs onto it, and it poisons it, and it disappears. This keeps repeating. But this is a video that he gave me, and he gave in his lecture, showing that if we stimulate our protection systems, they will destroy cancer cells. Now, I've produced a paper here. The second author here is a cancer surgeon for pancreatic cancer. And after the surgery, they give chemotherapy, but I was suggesting we give half-body low-dose radiation. And so the co-authors here are very renowned scientists, including Dr. Sakamoto in Japan, James Welsh, who's the president of the American Radiation Oncology Association in the U.S., I'm trying to get a study going on this, but people won't give me money to do the treatments with low-dose radiation. So I'm writing an application for a grant. Let's go on to another topic, the adaptive response. The low doses, and this is micronuclei in chromosomes. You probably know that every cell has got 2 chromosomes, and if you zap it with radiation, the chromosomes will break up. And so there's actually a natural incidence of micronuclei without any radiation. Those are the controls. And you can see it's a couple of %. And the low-dose radiation doesn't have any effect on micronuclei. But if you give it a big dose of 4 gray, and you can see that there's micronuclei, about 80–90% of the chromosomes have micronuclei. The interesting thing is if you give a dose 3 hours before of low dose, you actually improve the repair capability, cell repair. These are human cells. It was done in Chalk River Atomic Energy of Canada. And you can see that a preconditioned dose of 1 milligray, 5, 25, 100, and 500 milligray, given 3 hours before a challenging dose, all of them stimulate cell repair and reduce the number of micronuclei. So I thought you might want to see evidence that human cells can be, repair can be stimulated. Now we're getting to something that's close to my heart. The second author is Eugene Moore, this fellow, and Victor Hosfeld is the radiologist, and the Barbara Moore's physician is David Nadolsky. We did a study, not a study, we did a case report. We didn't know what to do in terms of treating this Alzheimer's patient. She was put in a hospice. Gene calls me up and says, how can we help her? And so I went through the literature. I saw studies on mice that low-dose radiation will work. The physician, David Nadolsky, said, I can't prescribe anything except CT scans of the brain. So we said, well, let's go with it. There's nothing else. So we did treatments. This is what a CT scanner looks like. CT scans are used for imaging, not for treatment, but we used it for treatment. And this is a paper we published. This is the second one. The first one was the case report. This is an update a year later. And this is a picture I took of her eating a sandwich. And then Gene and I were standing behind her, and I asked Barbara to look at the camera. And for someone who's totally non-responsive, non-communicative, she responded. I said, Barbara, look at the camera. So she picked up her eyes and looked at the camera. So we have restoration of appetite and restoration of, this is the only evidence I've seen of any improvement of Alzheimer's disease. There is no cure, there is no treatment. All you can do is alleviate symptoms. So I'm getting a study started next month. I've written a protocol. It took me 6 months to get people's attention. We're doing this in Toronto. We're treating 3 patients in a pilot study. And if we get good results, we're able to repeat the treatment was given to Barbara. I'm going to put in a proposal for big money to treat hundreds of patients and try and find what the optimum treatment is. So this is to show you that the first 3 treatments were the ones that produced the benefit. And then we kept giving her treatments because any treatment you give, there's always, it wears off. And so you have to keep stimulating. So we were giving boosters to Barbara, and she's still doing okay. This is 2 years. She was supposed to die within 6 months in a hospice. So she was kicked out of the hospice, and she's now in Alzheimer's care home, and he's taking good care of her. Now, this fellow's got Parkinson, as you know, you can see. And after he saw the benefit for his wife, well, it's the same kind of neurodegenerative disease. One is memory, another one is physical reaction. He was having tremors when he goes to sleep. He couldn't sleep, continuous tremors for hours. So he took the same treatment, and on the first night after the treatment, the tremors stopped during the sleep. He was able to get a full night's sleep. Not only that, after that he was able to reduce his medication to relieve the symptoms from 6 pills a day to 2 to 3. So these are the treatments he gets regularly, every 4 months or so. He gets a prescription from the doctor, goes in the clinic in Midland, and he gets a CT scan. And so he's got this under control. Now, there's people going around, David Brenner, telling us that cancer and radiological imaging. They're trying to scare people, saying don't get imaging, don't get CT scans because you got a risk of cancer. Everybody's terrified of X-rays. And here's 2 studies that were published last month in Canada. This is single CT scans. This is multiple CT scans. This is on mice. And these not only normal mice, these are mice that have a gene removed from one of the chromosomes, making them radiation sensitive and cancer prone. And so these mice were treated with CT scans, and they show more cancer latency. They pushed back the onslaught of cancer, and they increased lifespan. So these are two studies that contradict the scare. How am I in time?
You got about 15 minutes, Doctor.
Okay, well, this is a colleague of mine, Ludwig Feinendagen, wrote a chapter in a medical book called Therapeutic Nuclear Medicine, and he discusses the whole science of hormesis, low-dose radiation effects, and how it affects cancer, and we have to recognize upregulation of protection. That's basically what I've been talking about, is stimulating protection. And so there's a whole chapter on it. What he's pointing out here is all the studies ignore spontaneous endogenous. Endogenous is what happens internally. There's a huge damage rate due to breathing oxygen. And he says the endogenous rate is very high, and we should compare it with what's being caused by radiation. And so the number of cells, this will shock you, DNA alterations per day for each cell per day. Endogenous, this is due to reactive oxygen species in your own metabolism. Breathing oxygen. You get 1 million alterations in each cell every day. Each cell every day, 1 million alterations. And the number of double strand breaks, 1 in 10 days in each cell. Now, if you look at compare that with background radiation, well, it's 10 to the minus 2 and 10 to the minus 4. It's 100 million times less from background radiation, and 1,000 times, this is double strand breaks, 1,000 times less from background radiation. So everybody's causing fear from radiation when the big damage is being caused by oxygen. And it's not just a little bit. I mean, that's 100 million. And everybody's doing studies. Nobody mentions the damage that's being done by oxygen. They keep talking about radiation causing cancer. And he goes on here to say that low doses of radiation upregulate adaptive protection systems. We have two kinds of defenses. We have the fast defenses that operate immediately to remove toxins, repair molecules, remove damaged cells. And then we have delayed defenses. These are defenses based on 150 genes that last for a long time, months, weeks, years, and even a lifetime. And if you give these low doses of radiation, you can protect against renewed toxic impacts from both radiation and non-radioactive sources, like natural toxins. So you can get long-term protection with low-dose stimulation. And the adaptive responses are stimulated by 150 milligray. This is the dose that Dr. Sakamoto found with his mice study, where he injected the cancer cells in the tail vein, and he measured it against radiation dose, compared it with the controls, and he found that there was an optimum at 150 milligray. So that's an optimum with mice. We don't know what the optimum is with humans. We assume it's the same as the mice. You can't do studies on humans. So you do it on animals. So we got studies on mice, studies on dogs, fruit flies, and anyhow, this chronic or repetition gives you ongoing protection, increasing life and lowering cancer. Now, I want to show you something that's very interesting. Remember I was talking about plutonium in the lungs extending the lifespan of the dogs? Well, here's radiation to half body, okay? And we're looking at a tumor in the nose, and 54 days after the treatment in the trunk of the body, the tumor in the nose goes away, not in the beam. So this is signaling when you upregulate protection, it goes after cancers elsewhere in the body. So you can treat your whole body just with a local radiation, just like it was done with plutonium in the lungs. Now let's go to another study. We're ranging, I want to cover a lot of material here, and I've got a little bit of time here. This is studies we did in 1930s. Look how they did fluoroscopy for tuberculosis. They had an X-ray tube with a bit of shielding around it against a wall, the patient, and here's the radiologist sitting with the screen there looking at the lungs. So he's getting a dose to his forehead and his body, and the patient gets one dose much closer to the source, so he's got lots of radiation, but the radiologist looks at one patient after another. So he's getting a dose as well, and this is why you need radiation protection for radiologists. This was before they brought in all the lead shielding that we used afterwards. So this is a study that was done on a Canadian study on women who were examined with fluoroscopy for tuberculosis. They wanted to see if there was any breast cancer caused by the radiation. I mean, it's a natural thing to do. Everyone says that radiation causes cancer. So this is the data that was 50 years after the tuberculosis. These are the patients that survived. And I plotted the data. What they did was they averaged the low dose range to give you a linear model, straight line. But if you look at the doses the women were getting, some of them got a few examinations, so they got close to background. And then some of them got more examinations, so they got bigger dose. And you can see the increase in dose. It's a log scale. And it's not a straight line. It's not a linear model. It dips down, and you can see that in the low dose range, there's much less breast cancer death. And they're telling women that mammograms are increasing your risk of breast cancer death. It's false. It's only when you get to high doses that it causes increased risk. And the low dose range is beneficial. This is data that, it's not only me that's pointing this out, there's papers discussing this. Now, you've probably heard of the radium dial painters. Way back in the 20s, 1920s, there were a lot of ladies, a few men, 4,133 in the U.S., were painting dials with radium. And they pointed the brush with their mouth. They put the brush in their mouth to get a sharp tip. And so the radium that was on the brush was getting into their mouth. And of course, radium is like calcium, goes to the bones. So over the years, working for several years, painting dials, and then they decided they saw evidence of bone cancer developing. So there was a big study done by Rob Lee Evans at MIT, renowned physicist, on the radium dial painters. And it's an amazing thing he found. There was no incidence of bone cancer until you got to a dose of 1,000 rad. That's very high. And then above that, and this is a log scale, you can see factor of 10, another factor of 10. There's only about 30% of those that got the high dose ever got bone cancer. So there's something that's genetic here, but there's also a huge threshold. Now, if you took a linear model, why, you'd draw a straight line from zero, going up with bone cancer. But here you see very clear evidence of a threshold, zero bone cancer. And we're talking a lot of cases. Now, here's another thing that'll shock you. We treated 2,600 children. Sorry, not 2,000. Did I say 2,000? 2 million. 2,600,000 children with nasal radium irradiation. We put probes in their nose to treat adenoids, inflamed adenoids, shrink them. So remember I mentioned that radiation reduces inflammation? So this was an accepted treatment, 1945 to 1961, before the radiation scare. And these children were cured. Now the Navy pilots, in the early days there was no pressurized cabins or pressurized suits, suitable ones. A lot of pilots were susceptible to pressure changes as they flew up and down, hurting their ears. So they put radium in their nose to affect the eustachian tubes, the lymphoid tissue, so that the tubes would equalize the air pressure in their inner ear and avoid the pain. So this was 8,000 Navy pilots were treated with this. So you think, my God, there's going to be thyroid cancer, brain cancer, all kinds of cancer up the yin-yang. These are high doses they got. So this is the National Institute of Cancer report, 2003, and it explains how many were exposed. And the bottom line is no link to any disease. And this is after many years of study of both the children and the pilots have not confirmed a definite link between NRI exposure and any disease, not just cancer, any disease. This is another paper that I've written. I got 11 reasons to debunk a scare that thyroid cancer is caused by low-dose radiation. They did a study in Korea. When you do screening for thyroid cancer, you detect a lot of nodules due to naturally occurring growths in the thyroid. But if you look at the mortality, it's very low. People don't die very much from thyroid cancer, and when it is discovered, you can treat it with radioiodine. But if you start doing screening, examining people with ultrasound to look for little nodules, they did a lot of screening and they saw 15 times more thyroid cancer indications between 1993 and 2011 just by screening. And right after Chernobyl, what do you think they did? They went and screened all these children, and they saw lots of cancers, 5,000. They treated children, they took out their thyroids, they gave them now thyroglobulin. There was no need to do that because they never got that kind of radiation. It's a scandal. And this is published in the New England Journal of Medicine. This study was done in Korea not long ago. I mentioned, or we did, gas gangrene. Well, the early treatments, when you had it in, this is an agricultural worker, got his hand infected due to an injury in a farm with the tractors and whatever. Bones in his hand are broken, and it got infected with gas gangrene. So the normal treatment is surgery, cutting off amputation. But with low-dose radiation, you do 3 treatments of 50 rad. The first day, 2 treatments the second day, 1 treatment the third day. You actually cure the gas gangrene. It stops. And then you debride the damaged tissue, and it grows back. And then you reconnect the broken bones, and you save the hand. This was done in 1940s, 1930s. Now, as I mentioned, there are a lot of diseases that are affected by low-dose radiation. Well, this is a breed of mice that get diabetes. You can genetically modify mice to make them diabetic. So look what happens. You get the control group of diabetic mice, and then you can see what they look like on the bottom after a lifetime in natural radiation, natural background. And these upper mice, they got a low dose for the entire lifespan, and they live longer, and they look a lot better. So it works for a lot of illnesses. When you upregulate protection, it protects. So the damage is caused by oxygen breathing, aging, and if you stimulate your protection, you can deal with a lot of diseases. Here's another study, big report published in 2012 on these are the workers in Mayak. It's like Hanford in Russia. They had a lot of discharges into the river of radioactive material. People were drinking it. So they were looking at chronic radiation syndrome of a lot of these people, and they found that there was a threshold. Again, it's not linear. And most of the people who had exposures below 700 to 1,000 milligray a year had no higher cancer or leukemia than normal Russians living elsewhere. So how am I on time? Getting close? Is it safe to store nuclear fuel on site?
About 2 minutes, Doctor.
Okay, maybe I should jump over this. Basically, it is safe to store nuclear on site. Nobody gets hurt. It's all a scare. Conclusions, social concerns about nuclear energy safety. It's caused by a policy that links human-made radiation to cancer, risk of cancer. The radiation of 1950, the scare of 1956, it continues today. That's 60 years. Authorities are ignoring the beneficial evidence, beneficial effects. We need a policy change. to get nuclear energy accepted, and not only that, for medical applications. I showed you many applications where you can use low-dose radiation in medicine. The doctors don't want to do it because they think it's going to cause a risk of cancer. They have to justify every such thing. You got to go through ethics committee. The ICRP regulations that they had back in the 1930s are still valid today, and yet we've gone down several orders of magnitude in limits. So my recommendations are we need to discuss radiation health benefits, regulate, the bodies have to use the scientific method, the regulators, stop calculating low radiation-induced cancer risk using a linear model. We need a better public communication. You got to inform the people that the low doses are not harmful, in fact, they're beneficial. And there was a new announcement from the EPA yesterday saying that 100 rad of radiation is safe. I don't know if you picked up on that. We have a new leader there at EPA, Scott Pruitt. Things are changing there. You can raise the radiation level for evacuation from 20 to 700 milligray per year. That's safe. And look at this. American Nuclear Society, Nuclear News. Here's my article. And you may recognize the figure of Hiroshima, leukemia. So that went out in the September issue last month. And I'm sure there's going to be a lot of controversy because a lot of the members work in radiation protection, and now people are going to question what they...
Doctor, appreciate your testimony today, and Gene, I encourage you to, and I'll help you do this, to contact the chairman of the Health Policy Committee, because it would be just as relevant to them. But this is certainly a committee we went to school today, and gentlemen, thank you. We need to adjourn now in order to get to our next committee, and I need a motion to excuse those with absences. Vice Chair Lisinski so moves, without objection so ordered. The Health Energy Policy Committee is adjourned.